Maybe we should go.
So, morning everyone. Welcome to this international training course on respiration in the Mesopelagic Ocean, organised by the Scientific Committee on Oceanic Research Working Group ReMO. My name is Carol Robinson, and I'm one of the co-chairs of the working group, and I'm based at the University of East Anglia in the UK. As you can see from the panel of logos at the bottom of this slide, the course is supported by a number of universities and marine institutes around the world who have enabled the 14 instructors on this course to participate this week. So this first lecture of the course will explain why an understanding of mesopelagic respiration is so important in terms of constraining the biological carbon pump and ocean carbon storage, and therefore why we need to study it. How this is particularly important in a changing world with a future warmer, more acidic and deoxygenated ocean. But despite this immense importance, there are large knowledge gaps in our understanding. So the vast size of the Mesopelagic Ocean and its relative inaccessibility means that our knowledge and understanding is tightly linked to the innovations and challenges of the techniques that we use to measure mesopelagic respiration. So this lecture will therefore provide some context to the lectures that you'll get later today and the practical sessions that you'll get throughout the week, which focus on the advantages and limitations of a range of methods. Finally, I'll mention some ongoing international studies and future plans, some of which I hope you'll all get involved in. Excuse me. So let's start with a figure from the 2019 paper by Phil Boyd et al., showing the range of particle pumps which contribute to storage of carbon in the ocean. As you all know, the storage of carbon, which is out of contact with the atmosphere for decadal to millennial timescales, exerts a major control on climate by regulating the partial pressure of atmospheric CO2. This storage is governed by biological and solubility pumps, which maintain a surface to deep ocean gradient of dissolved inorganic carbon. The biological gravitational pump is driven by the settling of particulate organic carbon generated in surface waters through phytoplankton photosynthesis, and without which atmospheric carbon dioxide would be nearly twofold higher. Carbon is sequestered for longer than a year by particles that sink below the wintertime mixed layer, and for more than a century by particles that reach deeper than about 1,000 meters. As they sink, particles undergo a range of transformations that lead to the reduction in the flux or transfer efficiency of sinking particles, so that only a small fraction of the initial flux out of the euphotic zone reach the ocean interior. These transformations include the repackaging or fragmentation of the particles by the biota and microbial respiration of the organic carbon to carbon dioxide. Of course, it's a bit more complicated than this, and any budget would also need to include the physical export of particles via subduction caused by large-scale, mesoscale, or sub-mesoscale circulation, and the seasonal shallowing of the mixed layer. There are also biologically mediated processes which inject particles such as fecal pellets to depth through the diurnal vertical migration of mesozooplankton, which feed near the surface and respire and defecate at depth, known as the mesopelagic migrant pump, and the high latitude winter hibernation of copepods, catabolizing carbon-rich lipids that they accumulated in the summer near the surface, along with mortality at their hibernation depth, known as the seasonal lipid pump. But essentially, respiration is the process which reduces the efficiency of the combined carbon pumps, so arguably it is as important as the sinking flux, because knowing its magnitude and variability helps us to constrain our estimates of the storage of carbon in the ocean. So respiration is here in red because the balance of photosynthesis to respiration influences how much is exported below the mixed layer, and the respiration below the mixed layer influences how much of the particles are sequestered. It's particularly important to be able to constrain the biological carbon pump and thus carbon storage in a changing ocean for a number of reasons. First of all, to better inform policy development and decision-making in support of national and international net zero ambitions. This figure shows the annual assessment of the anthropogenic perturbation of the global carbon cycle from the Global Carbon Project, where the stock of oceanic organic carbon, although small in comparison with inorganic carbon, is a non-negligible component of the budget. And secondly, as part of the evaluation of the viability, efficiency, and ecosystem impacts of ocean-based carbon dioxide removal approaches, which are likely to be needed in addition to radical curtailment of fossil fuel emissions and land-based carbon mitigation strategies, if we are to keep global warming to below 1.5 degrees C by 2100. Some potential examples illustrated here from the 2021 IOC UNESCO report are protection and restoration of marine ecosystems and biodiversity, ocean alkalinity enhancement, macroalgal cultivation supported by artificial upwelling of nutrients, enhancement of microbial carbon pump to produce more recalcitrant dissolved organic carbon, and iron fertilization to increase phytoplankton production. So what do we know, and possibly more importantly and more interestingly, what do we not know about how respiration is affected by climate-sensitive parameters such as temperature, pH, and dissolved oxygen, and how respiration itself then affects the marine environment through the production of carbon dioxide and consumption of oxygen. So there is some empirical evidence of the positive relationship between temperature and mesopelagic respiration, with a Q10 of about 4 derived from oxygen consumption measurements. This is supported by a study in the Red Sea, which used backscatter and dissolved oxygen data from sensors on a biogeochemical argon float to derive seasonal apparent oxygen utilization in the mesopelagic zone related to temperature. However, a number of studies based on sediment track data to compare the remineralization length scales or the depth at which a third of the export flux remains with temperature are contradictory, with some showing that colder temperatures had longer length scales and others exactly the opposite. Respiration also plays a role in the decline of global oxygen inventory, where there remains a worrying 50% mismatch between model predictions of deoxygenation and actual measurements, with only about 5% of the global decline due to reduced solubility due to increased temperatures. Based on a review of the available literature at the time, climate-sensitive factors such as increasing temperature, increasing carbon dioxide, and increasing atmospheric deposition of nutrients all have the potential to increase rather than decrease respiration, so potentially leading to the dual problem of consumption of oxygen or deoxygenation and increasing production of CO2 or acidification. Devol and Hartnett in 2001 were some of the first researchers to show that dissolved oxygen may also be an important control on respiration. A synthesis of remineralization length scales estimated from export flux and deep sediment tracks by Steph Henson et al. Showed that mesopelagic respiration was reduced in low oxygen waters. So such relationships between respiration and temperature and dissolved oxygen provide parameterizations of respiration, which can be incorporated into predictive models. This model study of Cram et al. Investigated the role of temperature and oxygen on the efficiency of the transfer of particulate organic carbon from the surface to depth. They parameterized respiration in terms of temperature and oxygen, and then undertook model experiments to test the effects of them not including these parameterizations. And they found that removing the temperature dependence of respiration led to an underestimate of the difference in transfer efficiency of particles between latitudes, and removing the oxygen dependence led to an underestimate of the transfer efficiency, particularly in the tropical Pacific. A recent study by Henson et al. In an attempt to reconcile predictions of future export flux found that of the 19 global carbon models involved in the IPCC's 6th Climate Model into Comparison project, around half included a representation of the temperature dependence of respiration and half included a representation of the oxygen dependent, but only 5 of these models had both. Henson et al. Suggested that the magnitude of the bias associated with not including a temperature dependence was unknown, but potentially up to 20%, but that the likelihood would be that export would decrease due to an increasing temperature, increasing respiration rates. And they estimated that the bias created by not including an oxygen dependence of respiration could be positive, but that the magnitude is unknown, with the expectation, of course, that low oxygen would inhibit respiration, which would then lead to an increase in particle export in the future. So it's very clear then that there are several known unknowns in our understanding of respiration, and particularly mesopelagic respiration. And as part of an ongoing UK biocarbon programme, Stephanie Henson and her team from the National Oceanography Centre undertook a literature survey to try to assess the relative importance and the uncertainty associated with some of the processes involved in mesopelagic respiration. As you can see from the table at the bottom of the slide, they ranked particle type and composition, microbial solubilization and fragmentation, and aggregation by marine biota as those processes which were of high importance and highest uncertainty. They also undertook a survey asking the oceanographic community, and some of you may have been involved in this, what they thought were the most important knowledge gaps in our understanding of ocean interior respiration. And these are prioritized on the top right-hand panel as microbial solubilization, organic matter lability, and particle composition. So I think all of these threads of evidence show that respiration is important, but despite this importance, there are still major gaps in our knowledge. And of course, that's why you're all here, because part of the reason for these knowledge gaps is really the lack of a perfect method to measure respiration throughout the water column and across all scales from cells to ocean provinces. So the rationale for this present course is to discuss the range of methods that we have in order to determine mesopelagic respiration, in order to appreciate their particular advantages and their limitations, and therefore the appropriateness of using them to address specific scientific questions which span this vast scale of time and space scales. So we have a range of methods that can measure respiration from the cellular to the global scale. And this is important because the scales obviously cover increasing complexity of the biota, an increase in the complexity of the community, as well as the variability of depth, and also an increase in complexity of the substrate availability and mobility. So we'll try and position the methods that we're going to talk about this week along this scale. So they do cover these time and space scales. Some can only be used at a particular ecological or biological scale, and others overlap in their scales, allowing us to compare and contrast the methods at that particular scale. So in lecture 4 today, you'll hear about cellular fluorescence method based on the dye redox sensor green. In lectures 2 and 3, we'll hear about methods based on consumption of oxygen and the reduction of the tetrazolium salt INT. In lecture 3, we'll also hear about enzymatic methods, quantifying the activity of the electron transport system. In lecture 5, the determination of oxygen consumption using optodes on biogeochemical Argo floats. In lecture 6, biogeochemical methods to determine oxygen utilization. And in lecture 7, most importantly, the use of ecological models to both investigate and predict mesopelagic respiration. And this is why it will span the complexity in the community and the complexity in the substrate mobility. So let's take these fairly quickly one at a time. So you may have seen a recent publication by Munson and McGee, who used the fluorescence redox sensor green probe, an indicator of the oxidase reductase activity, to quantify cell-specific respiration in environmental samples. And this figure shows the relationship between RSG fluorescence on the x-axis and Winkler-based oxygen consumption on the y-axis. of samples collected from a range of open ocean and coastal sites. And we'll hear more about this method in lecture 4. The traditional method of determining epipelagic respiration is by measuring the consumption of dissolved oxygen in a bottle incubated in the dark for up to 24 hours. And I'm sure a lot of you have done this traditional method. And the decrease in oxygen can then be measured with either Winkler titrations or optodes. The bottle incubation method, although archaic, is relatively robust. However, the limit of detection over a 24-hour incubation restricts its use to the upper few hundred meters. The development of in situ respirometers, such as the IODA, and in situ sediment traps, such as Respire, enable the oxygen consumption of free-living and particle-associated microbes to be determined, without the challenges associated with collecting a sample from the mesopelagic depths and attempting to incubate it for a length of time at simulated in situ conditions of temperature, oxygen, and pressure. The advantages and limitations of measuring microbial oxygen consumption will be covered in lecture 2 and practical aid tomorrow. The traditional method of estimating microbial metabolism in the mesopelagic zone is to estimate the activity of the cellular electron transport system, or ETS. This assay was first introduced by Ted Packard in 1971 and is based on saturating the ETS dehydrogenase enzymes with their substrates of pyridine nucleotides to estimate the maximum activity, or the Vmax here, of the enzymes using an artificial electron acceptor, INT, which is then reduced to formazan. Deriving actual respiration from this maximum potential respiration requires an empirically derived relationship between oxygen consumption and Vmax, and we'll hear later about the variability of this relationship with things like species composition, body size, and food quality and quantity. The figure on the right is an early compilation of ETS-derived respiration rates through the water column, showing the attenuation with depth. And later today, we'll discuss more recent ETS data and their relationship with apparent oxygen utilization in lecture 3. To try to circumvent the limitation of requiring a conversion factor from ETS to oxygen consumption, Packard et al. In 2004 proposed the use of an enzyme kinetic model, which is based on biosubstrate kinetics to estimate respiration. They showed a good match between measured respiration and modeled respiration during the exponential substrate limitation and senescent growth phases of a bacterial culture shown here on the bottom left of the slide. Natalia Osma showed that this model, the V02 in the panel in the bottom, with rotifers and mycete shrimp under controlled laboratory conditions, and showed that it aligned well with oxygen consumption measured during incubations, here shown as R02, especially during food limitation. And you'll hear more about the advantages and limitations of this method in lecture 3 this morning. The classical ETS technique required the filtration of large volumes of water, transport of frozen samples back to the laboratory in liquid nitrogen, and relied on filters with pore size of 0.7 micron. So to try to circumvent some of these potential limitations, researchers from the University of Vigo developed an in vivo version of the method in 2009. Instead of killing the cells and measuring the potential enzyme activity, as in the ETS technique, the plankton cells were incubated and the production of formazan from the reduction of INT is measured. However, this also requires an empirical relationship to convert formazan production to oxygen consumption, with all the limitations that involved. The method had some major advantages in comparison with traditional oxygen incubations. It's more than 10-fold more sensitive than oxygen, and so incubations can be the order of minutes or hours rather than hours to days. And this means that variability can be measured on much smaller time and space scales. In addition, the plankton community can be filtered after the incubation, enabling the total community respiration to be attributed to size classes, such as a size class containing cells smaller than 0.8 microns, assumed to represent prokaryote respiration. On the right-hand side of the slide are mesopelagic data derived from the INT method collected from Station Aloha of Hawaii. So moving up to larger scales now, exploiting the proliferation of biogeochemical orgo flows in the World Ocean, Hansen et al. In 2016 highlighted the ability to derive depth profiles of respiration from oxygen measurements on a range of floats over an annual timescale. And we'll hear more about this in lecture 5 later today. We'll also discuss in lecture 6 the derivation of oxygen utilization rates from apparent oxygen utilization, from a knowledge of water mass age and dissolved oxygen concentration. Finally, today we'll discuss the use of ecological models to investigate the magnitude and variability of mesopelagic respiration and predict how it might change in a future warmer, more acidic and lower oxygenated ocean. So you can see that the range of methods to measure mesopelagic respiration relies on a huge range of expertise that spans experts in cellular microbiology, numerical modelling, biogeochemistry, and biological oceanography. And this scientific and technological expertise often does not reside in just one country. And since ocean carbon storage is really a global problem, then it requires international collaboration as part of the solution to the problem. So I've just now got some examples of international collaboration. One such is the UN Decade of Ocean Science for Sustainable Development, which began in 2021 and is due to finish in 2030, by which time we have the aim to reach a predicted ocean, the ability to import parameterizations of processes in carbon cycle into models that would then predict the ocean. So reiterating why it's important to better understand ocean carbon storage and thus respiration, on the left-hand side is a list of the state of play now, and on the right-hand side of the slide is where we need to be by the end of the decade. So success by 2030 would be that we were able to evaluate the efficiency of the approaches that might be proposed for marine carbon dioxide removal. So we not only need to cut emissions, but also potentially deploy removal processes. And these approaches will need standardized evaluation metrics, with which we can make decisions as to whether to go ahead with deployment, and if so, to then monitor efficiency. Metrics which can differentiate additional carbon drawdown and storage of carbon dioxide without concurrent releases in desperation. Henson et al.'s recent comparison of global climate model predictions of export flux suggested that they differed not only in magnitude, but also in direction. And Henson also highlighted our lack of knowledge of the activity of zooplankton, including the apportionment of respiration between microbes and metazoa. And there's the much wider question on the role of biology in the ocean carbon cycle and the numerous feedbacks within the system and how these could change in a changing ocean. So what are the mechanisms then of getting to where we want to be by 2030? Well, a lot of this, as I've said, depends on collaboration within international working groups and networks. And one such is the working group on respiration in the Mesopelagic zone, supported by the Scientific Committee on Oceanic Research, or SCORE, exactly the working group which is organising the training course this week. So our objectives include identifying and prioritising the knowledge gaps in our understanding of mesopelagic respiration, undertaking method intercomparison exercises, collating a global database, producing a best practice manual, and developing personnel through a postdoctoral mentoring scheme and this training course. A second mechanism is this integrative network with lead partners SOLAS, the Surface Ocean Lower Atmosphere Study, IMBA, the Integrated Marine Biosphere Research Project, CLIVAR, the Climate and Ocean Variability Predictability and Change Project, the Global Carbon Project, and IOCCP, the International Ocean Carbon Coordination Project, under the auspices of the Intergovernmental Oceanographic Commission of UNESCO. So a huge international network. And together we published this summary of the state of the art of ocean carbon research, which was published in 2021. And we're currently undertaking a similar exercise of what is our state of knowledge of ocean carbon cycling and what are the future knowledge gaps that we need to address. And that will be published in 2024. And you'll see that I've highlighted that one of the themes in this activity is the focus on understanding the role of biology and microbiology in ocean carbon cycling, including respiration and how this may change in the future. One of the UN Decade Programmes focuses specifically on mesopelagic processes, JETSON, or the Joint Exploration of the Twilight Zone Ocean Network, and this acts as a coordinator of mesopelagic studies. They run a monthly webinar that you're all invited to join. One of these national studies is MicroRespire, a project within the UK programme Biocarbon that I've just mentioned, and this aims to collate a global database of mesopelagic respiration to try to address some of the knowledge gaps we discussed today. So essentially, by forming this international community, we're pooling all of the data that people have promised that they will produce, and we can then integrate and investigate this large database. So then, I think I've persuaded you, I hope, that respiration provides a crucial constraint on determining the magnitude of the biological carbon pump, which is especially important in a changing ocean. There remain a number of known unknowns in terms of how respiration will be affected by increasing temperature, increasing carbon dioxide, and increase in oxygen, and the feedbacks that this then may initiate. There have been impressive recent developments in techniques used to determine respiration, and so it's really timely to interrogate these methods, reviewing their advantages and limitations, which is what we will do this week. I've mentioned a number of international networks who are working together to try to reduce the uncertainties in our knowledge of mesopelagic respiration, and these are communities that you yourselves can join. And I'm sure you'll hear much more about these exciting new data and new methods from the international experts gathered here this week. Thank you very much.